Good day, girls. Welcome back to the class. Today we'll do Science and Technology Part 2, the second chapter, Life Process in Living Organism Part 1. Now this chapter we'll be doing in two parts, and today we will do the second part. We have already finished the first part, okay, and today we'll be doing the last part of the chapter, that is cell division, an important life process. Now, cell division, an essential life process. We have learnt about cell in detail in the lower class. You have learnt plant cell and animal cell. You have learnt the difference between a plant cell and an animal cell. You know that a cell has a cell wall and a cell is filled with cytoplasm. And embedded inside the cytoplasm are cell organelles. Inside the cell organelle, the most important cell organelle is the nucleus. Other than the nucleus, the cell has organelles like mitochondria, endoplasmic reticulum, it has got vacuole, it has got Golgi bodies, and so on, lysosomes and everything. You have learned that. And every cell organelle has its own structure and its function. Today we are going to learn about an important part, that is the cell division. Okay, cell division serves as a means of reproduction in unicellular organism through binary fission. In multicellular organisms, cell division aids in the formation of gametes, which are cells that combines with the other cells to form sexually producing offspring. Now, as living things grow, some cell die or some gets damaged and need replacement. Some single cell organelles use as a type of mitosis as their only form of reproduction. In multicellular organism, cell division allows individual to grow and change by expanding the number of total cells. Okay, so you see how important is cell division. Now, cell division, there are two types. The first one is mitosis. Now, mitosis is a fundamental process of life. During mitosis, a cell duplicates all of its content, including its chromosome, and splits to form two identical daughter cells. When I say two identical daughter cells, means the cell undergoes division and it retains its number of chromosomes. Okay, and what is in meiosis, and the second type is meiosis. Meiosis is a type of cell division that creates egg and sperm cell. Now, meiosis we can say is also a type of division that results in 4 cells having half the number of chromosome of the original cell. Now, this part what I said, that in meiosis that cell division results in the formation of 4 cells having half the number of chromosome. We'll be doing at the end of this video, it will be well explained to you. Okay. So basic thing is you must know that cell division is an important property of the cell, then the significance of cell and the type of cell division. Now here let us discuss in general the difference between, you can say, mitosis and meiosis. Now in mitosis, here we can say it occurs in somatic cells or in stem cell. Now, what is somatic cell? Any cell of a living organism other than the reproductive cells. Okay. Now, what are the reproductive cells? You can say the male and the female gamete, the sperm and the ova. So, leaving out the ova, the cells of the ova and the sperm, all the other cells are known as somatic cells. And stem cells are special human cells that are able to develop into different cell type. This can range from a muscle cell to a brain cell. So, in all these cells, okay, mitotic division takes place, and meiosis occurs only in the germ cell, that is in the sperm and in the ova. Now, in mitosis, it takes place in two stages. One is karyokinesis and the other is cytokinesis. Now, you know that a cell contains nucleus. So, during this mitosis, it is the division of nucleus takes place. There are various steps to this. Okay, so when this division of nucleus is complete, we say karyokinesis is taking place. Once that is completed, then the division of the cytoplasm takes place. This process is known as cytokinesis. Whereas in meiosis, it takes place in two parts. One is meiosis 1 and meiosis 2. In meiosis 1, there is crossing over of genetic recombination. Now, you know that the cell inside contains chromosome and chromosome contains chromatid. When during this division, exchange of chromatid takes place, and that is when we say the genetic character is changing. Okay. Now, in mitosis, karyokinesis takes place in 4 stages. Now, we will be doing that after this. Okay. And in meiosis also, there are different stages in meiosis 1 and meiosis 2. You can just go through the notes. Now, the main difference between mitosis and meiosis is, in mitosis, the chromosome number does not change. For example, you have a diploid cell. Diploid means 2n. 2n undergoes mitosis and results in 2 daughter cell, 2 again diploid daughter cell. So 2n undergoes division to form 2n and 2n. Okay, so it retains the number of chromosomes. Whereas in meiosis, when say a diploid chromosome, okay, gets divided into n and n, where the number of chromosome is reduced to half. And the last point mentioned over here is the mother cell gives rise to 2 daughter cell. In meiosis, the mother cell gives rise to 4 daughter cell. This is a very important part which we will be doing it at the end. Now we will be watching a short video clipping wherein we will be explaining how mitosis is taking place in the cell.
You will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into 2 identical daughter nuclei. It is divided into 4 key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has 2 sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into 2 sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into 2 daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. the cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and becomes short and thick to form the chromosomes. In metaphase, microtubules of the spindle interact with the chromosomes. In anaphase, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubule becomes shorter. In telophase, daughter chromosomes arrive at the pole and chromatin starts to decoil, followed by the reformation of nucleoli and nuclei.
I'm sure girls, you must have watched that video and understood. Okay, so we have seen the different stages of mitosis. Okay, so here the notes are written for you about prophase, prophase, metaphase, anaphase and telophase. You can go through the video again and then go through these new notes. I'm sure you can understand. Okay, so that finishes off with mitosis. Okay, now let us see what is the importance or significance of mitosis. Okay, the main purpose of mitosis, that is it is helpful for growth and to reproduce or replace the worn out cells. I repeat, the main purpose of mitosis is for growth and to replace worn out cells. It helps in maintaining the same number of chromosome in daughter cells after division. It is also responsible for growth and development of multicellular organisms. It helps in repairing of damaged tissues. It also helps in the cell, you can say, in repairing the cell to maintain the proper size of the cell. So we saw how mitosis takes place. Okay, here you can go through the diagram. This diagram is there in the textbook also. Very important for you to go through these and understand the different phase of it. Okay, now we finish with mitosis and now we go with meiosis. Okay, now to understand meiosis, now meiosis, I said earlier, takes place in two parts, meiosis 1 and meiosis 2. Let us watch another video which explains meiosis 1 and meiosis 2.
Meiosis 1. In this module you will learn about meiosis 1. Meiosis is a nuclear division that occurs in the germ cells of sexually reproducing organisms during the production of reproductive cells called gametes. Meiosis is a reductional division that reduces the chromosome number of a cell by half. Meiosis involves a single round of DNA replication followed by two cell divisions, meiosis 1 and meiosis 2. The entire process of meiosis results in the formation of four haploid daughter cells with half the chromosome number as that present in the diploid parent cell. However, after fertilization occurs, the chromosome number gets restored. Meiosis 1 is the reductional division in which the chromosome number gets halved and distributed between two daughter cells. Meiosis 1 includes four stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. Let's know about the homologs before studying the phases of prophase 1. Each diploid cell has two copies of chromosome, one inherited from the male parent and the other from the female parent. These two copies, coming from different parents, are similar to each other and are called homologs. Now let us look at the different stages of Prophase I in detail. To begin with, let us learn about leptotene. During leptotene, the chromosomes condense and become visible. The zygotene phase is followed by pachytene. During pachytene, the chromosomal crossing over now takes place between the non-sister chromatids of the homologous chromosomes. This crossing over is what makes each individual unique and causes the recombination modules to be visible. As the crossing over completes, it marks the end of the pachytene phase. Diplotene is followed by diakinesis, which is also the last stage of Prophase I. During diakinesis, the chromosomes are fully condensed, the nucleolus and the nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase I is followed by Metaphase I. During Metaphase I, the microtubules from the opposite poles attached to the chromosomes at the kinetochores and the bivalents align along the equatorial plate with a random orientation. Either of the parental homologue takes up any site on the spindle. The metaphase I is followed by anaphase I. During anaphase I, the homologous chromosomes separate out and move to opposite poles with shortening of the microtubules. The sister chromatids, on the contrary, remain attached at the centromere. In this module, you have learned meiosis is a reductional division which reduces the chromosome number of a cell by half. Meiosis involves a single round of DNA replication followed by two cell divisions, meiosis I and meiosis II. Meiosis I includes four stages: prophase I, metaphase I, anaphase I, and telophase I. Prophase I is further subdivided into 5 stages: leptotene, zygotene, pachytene, diplotene, and diakinesis. During leptotene, the chromosomes condense and become visible. During zygotene, the duplicated chromosomes pair with homologues to form bivalents. During pachytene, the attachment of homologues to each other along their length, that is synapsis, is complete. In diplotene, synapsis brings to breakdown, the chromatids start to pull apart, and chiasmata are visible. By diakinesis, the chromosomes are fully condensed, the nucleolus and nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase I is followed by metaphase I, when the microtubules from opposite poles attached to the chromosomes at the kinetochores and the bivalents align along the equatorial plate. During anaphase I, the homologous chromosomes separate out and move to opposite poles with shortening of the microtubules, forming 2 haploid sets. During telophase I, the chromosomes reach the poles and nuclear envelopes are reformed around the 2 daughter nuclei and the nucleoli reappear. A furrow appears in the cytoplasm and the cell divides into 2 by cytokinesis.
Mitosis. In this module, you will learn about meiosis II. Have you ever wondered how the chromosome number in humans is conserved generation after generation? Well, this is achieved by a type of cell division called meiosis. Meiosis is a specialized nuclear division in which the chromosome complement of a cell is halved. Meiosis takes place inside the gonads of sexually reproducing organisms and results in 4 daughter cells, all of which are haploid. Meiosis involves 2 steps: meiosis I and meiosis II. Meiosis I is the reductional division in which the chromosome number gets halved. Meiosis I is followed by meiosis II after a very short interphase called interkinesis, during which DNA replication does not occur. Unlike meiosis I, meiosis II is an equational division. In this module, you will focus on meiosis II. Each daughter cell formed after meiosis I inherits only one of the two homologous chromosomes, and each homologue has two sister chromatids. Meiosis II involves the separation of these sister chromatids to produce haploid cells. Therefore, meiosis II is an equational cell division. As both the daughter cells produced from meiosis I undergo meiosis II, the final result of meiosis II is 4 haploid daughter cells. Like meiosis I, meiosis II can also be divided into 4 stages. These are Prophase II, Metaphase II, Anaphase II, and Telophase II. Prophase II is very short. During this phase, the nuclear envelope and the nucleoli disappear, and the spindle is formed with the help of the microtubules. In addition, the chromosomes recondense during the prophase II and become compact. Prophase II is followed by metaphase II, during which the chromosomes align along the equatorial plate and the microtubules attach to the kinetochores of the chromatids. Metaphase II is followed by anaphase II. In anaphase II, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubules become shorter. Anaphase II is followed by telophase II, which also marks the end of meiosis II. During telophase II, the nucleoli are reformed and the haploid nuclei get enveloped in membranes. In addition, the chromosomes decondense and the spindle disappears. Division of the cytoplasm, cytokinesis, results in 4 haploid daughter cells. Meiosis plays a significant role in the life of sexually reproducing organisms. It results in gamete formation and helps conserve the chromosome number in sexually reproducing organisms. All the 4 gametes produced are genetically unique as they are produced by the crossing over of homologous chromosomes. The genetic uniqueness of the gametes is further attained by the independent assortment that results in each daughter cell receiving either homologue from each chromosome pair. Let's summarize what you have learned. Meiosis is a specialized nuclear division in which the chromosome complement of a cell is halved. Meiosis involves 2 steps, meiosis I and meiosis II. Meiosis II has 4 stages, prophase II, metaphase II, anaphase II, and telophase II. Prophase II is very short. The nuclear envelope and the nucleoli disappear, and the spindle is formed. The chromosomes recondense and are compact. During metaphase II, the chromosomes align along the equatorial plate, and the microtubules attach to the kinetochores of the chromatids. In anaphase II, the sister chromatids separate out and move towards the opposite poles. During telophase II, the haploid nuclei gets enveloped in membranes. The chromosomes decondense and the spindle disappears. Cytokinesis results in 4 haploid daughter cells.
Meiosis I.
I'm sure, girls, you have understood the video and understood how meiosis I and II takes place. So this is the diagram related to meiosis part 1, and this is part 2. You can closely have a look at the figure and try to understand. Okay, so you have seen how there are 2 stages of meiosis, that is meiosis I and meiosis II. By meiosis, from one diploid cell, 4 haploid cells are formed. In meiosis, crossing over between non-sister chromatid of homologous chromosome occur. This results in genetic recombination. The 4 daughter cells formed after the completion of meiosis are genetically recombined and not exactly alike, and also not exactly similar to the parent cell. Now, spores and gametes are formed by meiosis. Meiosis helps to restore and maintain the chromosomal number. Now in meiosis 1, if I have to tell, the homologous chromosome undergoes crossing over and hence there is a genetic recombination. The homologous chromosomes are divided into two groups and from these two haploid cells are formed. Prophase I of meiosis, if you see, prophase I of meiosis is a lengthy phase which is subdivided into 5 phases. Okay. Now in meiosis 2, which is similar to mitotic division, the two haploid cell that are produced in meiosis 2 now undergo further division forming 4 haploid cells. Let me explain that. Okay. Now suppose, now we are talking about meiosis 1. Okay. In meiosis 1, we begin with a diploid cell, that is say 2n. Okay. Now this is a diploid cell. This undergoes meiosis 1. Now here what happens? The division takes place and two daughter cells are formed. Now if you look at this, okay, if it begins with 2n, it undergoes meiosis 1 and here 2 haploid, 2n gets converted into n and n. This process is known as the halving process, where the number of chromosome is reduced to half. Now after the completion of meiosis 1, where a haploid, okay, now diploid is 2n, haploid is n. So after the completion of meiosis 1, it undergoes meiosis 2. Now meiosis 2 is almost similar to mitosis. Okay, here this is meiosis 2 now. So here each daughter cell which is haploid, okay, now gets divided into again 2 haploid cells. Okay, so here the second part of meiosis 2. The second part of meiosis 2 here is similar to mitosis. In mitosis, I said in the beginning, during mitotic division, the number of chromosomes are retained. So in the second part, you can see again, you can see there are 4n formed over here. So in short, I can say, we can summarize that, okay, thus we can conclude that each diploid cells form 4 haploid cells. So here we begin with a diploid cell and then we finish off forming with 4 haploid cells. This is how meiosis 1 and meiosis 2 takes place. I'm sure you have understood this concept. Now again to revise with the importance. Why is meiosis very essential? Cell division. Now meiosis is responsible for the formation of gametes. They are responsible for sexual reproduction. It activates the genetic information for the development of gametes. It also maintains the constant number of chromosomes by halving the same. So I hope you have understood mitosis and meiosis, which is a very important process that takes place during cell division. Thank you and have a great day.